I look at mitochondria as like the brain of the cell. You know, it's like insofar as it's where the information is coming from, being processed. Uh, Although probably the microtubules are more closely associated with storing that information long term, because the mitochondria is so dynamic. But that particular configuration of the DNA molecule within mitochondria, uh, well, for one thing, that gives it a very strong morphic resonance, if you will, with the first life forms, because the first life forms to use DNA had circular DNA, because uh, DNA strands that are open-ended, those ends are open to chemical reactions or attack. So it destabilizes the DNA. The most stable configuration for it is this plasmid structure. So it probably shows that's probably one of the strongest links, taking us back to like the origin of life within us still. And then, um, you know, it becomes extremely interesting when you look at different configurations of the DNA molecule, and how that affects those delocalized electron orbitals. So when you're talking about just a linear strand of DNA, like in the chromosomes in our nucleus, uh, by and large, um, certainly within the euchromatin, the protein coding segments, um, it's the pi orbitals are just stacked one on top of each other, and they're entangled, quantum entangled, and oscillating together. Now, if you look at the genome, though, almost 60% of it is what's called repetitive DNA elements, variable nucleotide tandem repeating elements. And this is why it's called junk, because if the DNA encodes information via the nucleotide sequence, if the nucleotide sequence doesn't have any variability to it, if it's just a repeating number of nucleotides like G, C, G, G, and that just repeats like thousands or millions of times, that can't encode any information, certainly not protein coding information. But if you look at what it actually does, that 60% of the DNA is very dynamic, and it's actually functioning in like a 4D space. So it's larger geometric configurations that it forms that are the most significant. So it can start higher order geometrical configurations. And with those kind of configurations, one thing, I mean, it looks just like electrical components, you know. So this is actually like ultra compactified DNA. It forms this, torus. But you can also see where DNA can form very specific three-dimensional configurations, and this is happening within the biological system. So endogenously in your cells, where it's that so-called junk DNA, that repetitive DNA, it's not encoding in the information in the nucleotide sequence. It's encoding bonding patterns, and so you can get very interesting bonding arrangements, even Uh, Four-stranded DNA, that's very typical of the telomeres at the ends. But so when that happens, it's producing a novel electron orbital configuration. And so the light that's going to be encoded there or released there is going to be different than the normal single strand. Going back to the mitochondria, that circular DNA, it's going to have a different kind of electron orbital configuration. So it's going to be storing and accessing information that is different from much of the nuclear DNA.